It's an honor to be here. It really is. Thank you to the Broad, to Anna, to the Tremere Foundation for this wonderful support. As a relatively newly minted member of biopharma, I'm here to share both good news and bad news. Bad news, biopharma really stinks at making drugs. We get it done eventually with enough money, I think, as Anna pointed out, but we're really terrible at it. The good news is we believe there's a really good solution, and that's in the patients themselves. They are part of the problem that we're trying to solve, but they're part of the solution. And so what I'm here to present today is not so much the science, though we're going to dive into it a little bit. It's really how do we make this infrastructure work for everybody? And so our strategy at Unravel is patients first. That is the problem. That is the North Star. We're singularly aimed at that, and let's work together to figure out how we get there. And we're doing this through a very heavy partnering approach. So here is traditional drug development in a very abstract sense, but you can see that there's a target. This usually comes out of academia. Mouse highlights any sort of preclinical models, cells, animal models, et cetera. And then you get into the critical question, does it work in patients? This is what we've been trying to do this whole time. And this is where my earlier bad news comes in. We're terrible at this. It takes years, if it's even going to be successful. On average, a new target has about a 98% failure rate in terms of this approval process. So this doesn't work. And this is for non-rare diseases too. But when you get to rare, and especially ultra-rare, this just doesn't work, right? So what can we do? And what, kind of digging one layer deeper, what's the problem? Our thesis is that people are trying to treat disease, when really we should be thinking about treating patients. Right? Diseases are not the same as patients, and so a shout out to the Cleveland Clinic for these really helpful graphics, but they represent the generalities. They don't represent individual patients. So again, what we say is it's the patients, not the disease, that responds to the treatment. And here's actually a picture from our office that reminds us this is why we're here. And actually, Luki is on there as well. And so, this is not a new concept. I would love to be able to take credit for it, but it actually goes back to the 19th century with William Osler, so the father of modern medicine. Basically, the good physician treats the disease. The great physician treats the patient with the disease. And I think we've lost track of that along the way, and so we're trying to bring that back. And part of it is because the technologies today enable it, and part of it is because the patient advocacy groups are so freakishly sophisticated and motivated that now we can do it, right? So it's this kind of, it takes a village, but even more complex concept. And so what we did at Unravel is we have this patient-driven drug development. And there's, people pay lip service to patients, and every pharma company has a page that says, oh, we're patient-centric. That's great. But have you actually worked with these groups? Have you worked with the patients, right? And that's sort of the question. And so, again, why do we start with patients, right? And to some extent it's obvious, but to some extent there's really deep biology. And the scientific problem is a really big one. And I think Anna mentioned this really nicely in the beginning. We have no clue what half of the pathways are doing. You have these proteins, genes that have been identified, that's great. No idea what they do, or maybe we know one thing out of 100. The reality is that what constitutes a patient, right? Now, not just a human, but just a patient specifically. Sure, genetics in the case of genetic disorders, the environment, this includes diet, medications, et cetera, and time. And I think Ada and Nathan really nicely alluded to it. When is a really critical question to ask, not just what and how and who. And so what we did, and we spun out of the Harvard ecosystem, so just across the water here, out of the Wyss Institute, and there we had this insight that these complex biological networks drive clinical function. And here's actually a real example of one of our networks. And the idea is that these interactions, as opposed to any one component of this, drive patient outcomes, right? It's kind of the sum of all the parts. Now, if you have tried to drive through Boston, for example, just coming here today, there was a malfunctioning traffic light. Turns out that backed up the traffic onto Sturo, which backed up onto I-93, which who knows what it's doing, right? So one traffic light can have these vast effects. That's a network problem that you encounter every day, especially in Boston. And so let me walk you through how we do this. So obviously this teeing up to a computational example, as you might imagine, but the idea is that you have this healthy network, and again, just completely arbitrary. This is actually from a Rett syndrome patient I'm going to talk about later, but don't worry, the yellow lines are just for show here. You have some sort of healthy interaction. This is what normally should be happening. And there's a problem. There's a gene maybe that's mutated. There might be an infection that's causing something. Who knows what it is? In this case, it's actually the MECP2 gene that's highlighted here. There's some problem. There's a mutated node, and you heard this nodal biology is really kind of getting at that. So great, it causes some disruption. It's that malfunctioning traffic light, right? So how do we fix it? Well, you could fix the traffic light, but you can also fix anything else that rebalances this network. And that's the really great opportunity in biology. It's a mess. And so why focus on just one thing, right? You can take any of that mess and use it to your advantage. As unfortunately every oncologist knows, cancer cells are really good at finding these alternative pathways. So why can't we use that for good? And that's really what we do at Unravel. And essentially what we do, we're forecasting how patients respond to therapies. Again, it's about the patient, not the disease. So we have patient RNA signatures from actual individual patients that we work with on a daily basis. And we have drug RNA signatures, and not unlike what Anna showed with the visual signatures, these are really transcriptomic signatures, but from the perspective of these networks. And then we go through this magic, and we ask, will it help this patient, right? Lukey, for example. Can we treat all the patients within a particular disease, or however you want to look at it? The answer may be no, but you can treat some of them in one way, and you have to find something else for the others. Are there better treatments? So repurposing might be a great place to start. We agree, but we're also developing new drugs that are now tailored to the mechanisms and targets that we discover. And one thing I want to mention is that also looking at the adverse side of it, right? So you make all these molecules. One of the biggest drivers of costs are all the failures, toxicities, lack of efficacy, and all that. But toxicities are, put patients at risk and are just bad business. So just, there's no reason to not avoid that if you can. And so we see all of that in a pretty clear way. So we bring this function-first approach to drug discovery, and we have sort of these three components. One is BioNav, and it's really this Google Maps for health, this network biology of human health, coupled with an in silico compound library, about 40,000 molecules. Now, all these are purchasable. You could say this is repurposing. To us, they're discovery tools. And over time, families and foundations like the ZTTK Sunshine Foundation came to us and asked, well, can we just use those tools for the ones that are actually approved for patients? And the answer is yeah. We're not a repurposing company, but we're trying to probe the complexities using these molecules. One thing that we do as an intermediate step, we developed this concept of squishyware. It's not hard, it's not soft, it's squishy. These are tadpoles. They are actually very squishy. And we can do sort of a kind of computer vision type analysis, like again with the cells that you saw earlier, but with living, moving tadpoles that are, again, mutated with the various genes. And we can just ask, does it help that, rescue that model? But then really where we're at, again, it's patient-driven and patient-focused drug development, or the clinical studies. Does it actually work in a patient? And so what we do, we use existing drugs to ask that question, because you could administer those today, not in 10 years, right, with potential other toxicities and other things. Yes, they may not be ideal. They may not get you the 100% of the efficacy you're after, but if you can get 50%, one, it proves out your mechanism, that you can now invest those resources for a new drug, and it might help patients today in the meantime, right? So you don't have to wait 10 years for the perfect drug and have nothing. You might have an intermediate step in between. And so does it work? Well, yes, it works, even to our surprise, quite well. We've actually shown this actually during the lovely pandemic that we had a few years ago. We showed that we can predict the function of specific statins in this case, we use for cholesterol control. You predict specific statin impact on survival from COVID. So these are patients who are getting statins because of their hyperlipidemia anyway, and we showed that specific molecules, but not the class as a whole, were actually therapeutic. And this was death as an endpoint, so pretty cut and dry in terms of outcomes. And we were statistically significant. We had a false negative, which is a good one to have, but overall much better than your 2% success rate. We had an 80% success rate in predicting clinical outcomes. And so part of the challenge here is really this infrastructure and how do you work with patient groups, how do you partner? And I mentioned that families started coming to us, and so we actually had to develop a new business model. And we set aside the one that we started out with about 4 years ago before spinning out, And we said, we really need to offer these services, even though we're not a service company and we're not a repurposing company. And so we started the RareShift service platform. And really, this is just sort of cost-based drug discovery for nonprofits. It's at rareshift.org. And there it is drug repurposing. That is the goal. That is the message. That is the goal. For us, it's a stepping stone towards the novel drug arm. Essentially provides clinical discovery and validation, and then the partnerships build in the process. And we become a partner to the foundations or the families, and they become our partners in the discovery and validation process. So it becomes a win-win. And what we've been able to do is actually using nasal swabs. So again, before the pandemic, this would not have been possible, to collect a few cells on a COVID test kit, essentially, and use that to extract RNA, run it through our BioNav platform, and get insights on the brain, because most of what we work on are neurological diseases. And then we can use this AI platform to figure out how do we potentially fix this patient, what's going on, how is patient 1 different or the same as patient 2, and so on. And what's really exciting, we can work all around the world. So we've actually sampled patients from the remote outback. Took 5 weeks, but we did it. We can basically ship these kits all around the world at room temperature. And so what we have started to do is see the evolution of these diseases into truly what we call therapeutic response groups. It's not about how you got there, it's how you fix things, right? And so that's where these TRGs, therapeutic response groups, come in. And so this is actually SYNGAP1 is another one of these neurological disorders. If you take a look at this rainbow matrix, there is no pattern. We tried. There's nothing really clear. Different ages, different mutation sites, genotype, phenotype doesn't really exist. But what we start seeing in terms of drug response are 3 groups. And they actually correlate really beautifully. And those patients are actually quite different from each other once you look at them from that perspective. And so now we're trying to take these really complex populations and reduce them to something more manageable. And so just to show what we've been able to do on the biopharma side of our company, we work on Rett syndrome. It's, again, one of these neurological neurogenetic disorders, one of the more common ones. This is not rare, and there's been one approved drug on the market. And we actually use that as a benchmark, and we basically did the same process, collect data from patients and from other sources, run it through our platform. Here's actually the mutation, but we found a target somewhere else. Doesn't matter what caused it, but if you can engage this target right there, right? How we got there is the magic, but we show that if you can actually engage the target right there, it fixes the network. And that was the real insight. And so now we actually have a new drug for this, but we're not going to develop it until we show that this works with an existing drug, because that's how we can test it today. By the way, the existing drug, verinostat, formulation in Ravel 1, outperforms the one that's currently on the market dramatically, especially in the later stages of the disease. And we're actually starting 2 clinical trials in Rett syndrome. And then you may ask, does it actually work? Well, this is the beauty of using existing drugs. There's actually a family with a boy with Rett syndrome. So boys are really rare. They tend to not do very well with this disease. And they were really looking for options. Their clinician was looking for options. And so long story short, we identified verinostat actually to work for that patient computationally. And the clinician felt that it was appropriate for her to prescribe it to the patient. And sure enough, extremely low doses. And this is a cancer drug, right? So keep in mind, this is not a supplement. The sleep quality improved pretty dramatically. Normalized blood metrics. This is an objective measure of improvement. And then improved motor and cognitive function as assessed by various clinical professionals. And now we're actually, the boy's still on it. This has been on it for over a year at this point, continues to improve. And we're actually layering additional therapies on top of that. I mean, the clinician is, but with our data. And what we're doing now, this was really exciting to us, because it's saying, look, this really can help even reverse this extremely severe case. We're actually starting 2 clinical studies, one in Rett syndrome in Colombia. So I joke that we're bringing white powder to Colombia for a change. It's a really fantastic place. We go to where the patients are. There are 15 patients all being treated by a single clinician, single site study, really fantastic place. And they have never been able to participate in cutting edge research like this. And so this is what we're doing. We're actually using oral liquid verinostat, validating our target, right? Now there's a repurposed candidate. We have orphan drug designation for it in the US. We will bring it to the US. We also continue with it in Latin America. But then we're actually building Ravel 2, which is this new optimized drug specifically for Rett syndrome and several other diseases. But we asked the critical question, does it work before we commit $10 tens of millions of dollars to developing a new drug. And it gets even better. So the question is, there's 15,000 rare diseases, or whatever the number is, a really big number. How do we tackle not one at a time, but how do we parallelize it? And so we've actually identified these therapeutic response groups to span diagnoses. So there's Pitt-Hopkins syndrome, which is not Rett syndrome. It's a different gene, different disease, with some similarities. But they're actually sponsoring a clinical study because we predicted that it would actually be quite effective, predicted that it would work in mice. Lo and behold, it worked in mice. So now we have to go to patients. That's the only way to answer that question. And overall, we have about 50 different diseases in our portfolio. As you can see, they usually start with this BioNav screen. Some of them have tadpole models that get developed. Some don't. Some have mouse models that get developed. Some don't. We work with Jackson Labs as well. Where people are really headed is the clinical part, right? Does it work in actual patients? I mean, that's the ultimate question. And so all these are off-label. I should note that we do not treat patients, but we generate research use data that can be used for clinicians to evaluate as they see fit. One thing I want to announce here today, so we actually have not announced this yet, so this is the first time. We just signed a very key agreement with Combined Brain. So Nathan and Ada mentioned them. They're an umbrella organization that really spans about 120 different patient advocacy groups, encompassing a little bit more diseases than that. Some of them have actually more than one falling under them. And essentially what we're doing is taking our really patient-first approach, RNA-driven approach, and applying it to not just the 50 disorders that we already work with, but another 120. Right? So this represents really a landmark partnering in the space. Rare disease, most of these are ultra-rare diseases, ultra rare. I mean, one group has 6 patients documented, right? So these are, talk about a challenge of scaling to commercial opportunities. And this is the type of partnering that I think we envision, that we, thanks to all these fantastic patient advocacy groups, are really trying to change how we do drug development. So with that, I want to thank you for your time, and again, drug development driven by patients.